The idea will be to give you an introduction to what we do here at the MRC T, give you a feeling for the kind of challenges that we face. Oh my goodness, there it is. It's come alive, and also then give you a good introduction to a small project that we've done here to illustrate some of the challenges that we face. So moving on to the next slide, hopefully we can go in. Yes, we can go now. So the remit of my organization is actually to do target validation. So the MRC T originally spun off from the MRC, which is the Medical Research Council in the UK. The idea there was rather than just doing a technology transfer in the traditional sense of the word technology transfer, where you would be looking at looking after IP, the MRC T decided also to look at how you would be able to move projects or targets, drug targets that have been discovered in academia, and do a bit of de-risking so that you will be able to sell these targets on to big pharma. And when I say sell, I'm not talking about actually the money, but also encouraging big pharma to work on more challenging targets. So we pride ourselves as a bridge between the academia and pharma, and I think I'll come back to this theme as we go through the presentation, that actually our job is not necessarily to compete with big pharma, but our job is to augment and to enable. So the MRC T Center for Therapeutic Discovery is involved in many things, and I've just given you on this slide a lot of the things that we do. So we do assay development, hit generation, lead discovery, and lead optimization. And we also work on antibodies and humanizing antibodies and antibody engineering. So we do cover a lot of areas in terms of what we do. But the talk today will be focusing on small molecules and what we do in terms of small molecule discovery. I've got a slide here that just gives you a feeling for some of the portfolio of the targets that we have. As you can see, some of these are not the easiest targets to work on. So we take targets that are quite early, i.e. targets that are coming from academia, and here I've got a few examples of targets that are actually in process or have been worked on in the last 2 years at the MRC T. Just to give you a feeling for where we hope that all these things end up, these are some of the therapeutics that have ended up on the market at the MRC T, and if you want to see a little bit more and to hear more about us, please visit our website at mrcttechnology.org at the bottom of the slide.
Okay. Chido.
Thank you.
That's right. Yeah, I jump in here. Just excuse me, because before Chido is actually starting to dive more into science, we would like to be able to pitch best the level of explanation, and Chido would like to get a better feeling for who he is talking to. Therefore, we have the possibility of actually doing quick polls here. If you could be so kind to let us know who you are, what your environment is, we'll do 2 quick polls, one after the other. Please, if you'd be so kind to click, I fire up this now. So this first question is about your work environment. If you could be so kind to just quickly pick, yeah, the participation rate is going steep up. Almost half of you have already clicked, so never fear, it's only a click away, and it helps us pitching the questions better. Yeah, it emerges roughly. I wait another 5, 4, 3, 2, 1 seconds, and I close this so that you guys can actually see what's going on. So half and half, pharma and academic. That's interesting. We never had this before, actually. So whoever is from industry mostly is a comp chem. Whoever is from academia mostly is a med chem. That's good to know. Chido, you recorded this in your brain, probably.
That's great, yes.
Excellent. Thanks for that. And just one quick second poll here so that we get a better feeling for your responsibility. Is it best described as bench chemist, project manager, student postdoc, computational expert, or something other, if you could again be so kind to quickly click so that we get a better feeling for who's in front of us. Okay, yeah, getting up contribution. Okay, 5, 4, 3, 2, 1, 0. That's again a mixed bag here. Lots of students and postdocs, that's great. You can learn from giants here, computational expert a third, and quite a few bench chemists again amongst them here. Okay, thanks very much everyone. That was of great help. I'll dim out these results and hand over back to Chido to dive into scientific stuff now. Thanks very much.
Let's move on to the next thing. So I wanted to give everybody a feeling, especially the guys who do CompChem, what kind of things we do on a day-to-day. So at the MRC T, as I described before, we've got new targets. And normally these new targets, depending on the level of validation and the level of structural information or the level of O2 compounds that we have, etc., we would prosecute them in a different way. But this is just a rough schematic of what we will do. So basically at the MRC T, we've got screening sets, and we've got a diversity set, which is about 200,000 compounds. We've got an index set, which is representative of that diversity set. And thank you to my colleague Chris Birchall for designing this diversity set. That gives us a feeling of the kind of diversity set by screening the index set. So you don't have to screen the whole set when you're doing an HTF. We've also got some focus sets here. We've got kinase sets, ion channel sets, and peptidomimetic sets. And in the last 3-4 years, we've been going towards fragments and annotated sets. And as you can see, all these sets are kind of ready for biochemical screening, biophysical screening if it's fragment sets, or phenotypic screening if you're looking for specific phenotypes that you want to screen. So we are quite agnostic about the area of work in terms of disease area. And our targets, because of the nature of the targets, we don't always work on specific families, but you'll find that a lot of our work has been done on kinases if you look at the literature. But however, where does CompChem sit in terms of what we are doing? So CompChem sits here in terms of trying to influence how the libraries are designed. So I've highlighted some of the software that we use, fingerprint similarity, feature tree similarity, to try and understand what is in our libraries. We use ligand-based virtual screening, docking, using both feature trees, fingerprints, or docking to propose some screening sets for screening. We also are involved in terms of generating follow-up for hit validation activities. So if we found something as a hit from an initial screen, we would then hunt either within our own libraries or outside of the MRC's libraries for analogs. And here we find that in silico tools are very useful, e.g. feature trees or fingerprint similarities. And then as part of our lead development processes, we would use either ligand-based drug discovery or structure-based drug discovery. And this includes things like homology modeling, docking, feature tree scaffold hopping, as well as FlexS if you are doing ligand alignment. So that gives us a kind of feeling for what activities are done at the MRC T in terms of the computational support for projects, and in terms of the nature of the projects and the nature of things that we normally work on. So here we go. I was going to use this particular presentation that's coming up now to illustrate this particular project, to illustrate, oh dear. So we're having problems again. if you can jump in and let's see, maybe I might have to fire up an email to you, but
yeah, or you save it now as a PDF maybe, and then open just a PDF if that makes sense to you.
yeah, that might be the way to go. Just one more time. It looks as if it's not a. We experienced something like this before, Marcus.
No, never actually. And especially since we tried all this out before, of course, being professional on that side.
Okay, that's it. It must be the way we've installed something. Okay. So the example project I wanted to give you is how we managed to take small molecule, small fragments from really poor hits in terms of low micromolar hits, and how these were developed to nanomolar inhibitors of PAICS enzyme. Human PAICS enzyme, I'm going to describe the target now. The target is for breast cancer, and basically this target is known to be involved with metastasis. So if you hit this particular target, you stop metastasis. In fact, although there is a lot of pre-screening these days for breast cancer, a lot of people are still dying from breast cancer in a year. In fact, more than 500,000 people die in a year in Europe and the UK. So this is an interesting idea of using this particular target to go for cancer. And what was the information that was available to us at the beginning? So at the beginning, this was considered an attractive novel drug target because it was expressed in cancer cell lines. If you did an RNA a knockdown, you would abolish metastasis, and it also looks as if this gene is highly expressed in cancer patients who have shown poor prognosis. So the PAICS target itself is a bifunctional enzyme which is involved in de novo biosynthetic synthesis. So I've given an illustration of the pathway here, and these two steps are the two steps that are catalyzed by PAICS. Okay, so just to use a shortcut, we call this air, and that is converted to K in the carboxylase part of the protein, and that is taken to Cy-K. And this particular step number 7, is driven by ATP. And so in the beginning, this is what we knew. And if you look at the graphic at the on the right-hand side where I'm mousing over here, you can see that actually CRISPR knockdown has been used to validate the target before we worked on it. PAICS itself is expressed in other cancers, but highly expressed in other cancers, but it was particularly so in breast cancer. So the initial plan was easy, right? So you just get the MRC T library. At that point would have been 125,000 compounds. We would augment it with collaborators library. So this is an AZ library which we have access to. Would screen these libraries in-house and end up with hits, and these hits would then be the basis for our crystallography studies, come up with crystal structures, use this information to do hit-to-lead optimization, and this is easy. I mean, before you know it, you've got your highly potent inhibitors, and you can put them into functional cell-based assays, and then confirm mode of action. And at this point, because of the nature of our work, we would have come up with a small molecule, which is a tool compound that is useful for validating our target. So I'm going to move on quite quickly to avoid the interruptions. So the assay development was done here at the MRC T, and we managed to come up with a good way of screening especially focusing on the depletion of ATP to ADP. So, but after doing the HTS, there were no hits. In fact, the only hit compound that came up is shown in here, is shown down here, and this particular compound is it had poor potency. So we're talking about 15 micromolar here. It had if you're a chemist, you'll be looking at it and going, where do you go with this particular compound? So this will be called an ugly structure if you're looking at it from a med chemist bench. But however, this was useful as a tool compound in terms of using a control for the assays, etc., etc. But we didn't know what else to do. In fact, a little bit of chemistry was done around this particular core, i.e. removing some of the biphenyl ring here, making sure that you just have one benzene ring at the bottom and you lose all activity. If you modify any part of the compound, you seem to lose activity. So there was no. It looked like we're in a bit of a cul-de-sac here. So why not fragments? So, however, normally when you're going to prosecute a fragment drug discovery project, you have to have confidence that you're going to be able to get crystal structures. And I will explain how we approached the problem. So here we go. We threw our library of fragments, which is at that point we took about 1,000 compounds in our fragment library, and we managed to get a reasonable hit rate. About 11% of the library came up as hits. And this was on the biochemical assay, and 15 of the hits that came up at the top. On a biophysical assay, and 15 of the hit that came up gave an IC50 that was readable. These were taken to Biocore, and using that particular compound as a control, we're able to show that actually we have a good way of immobilizing the protein on the surface of our Biocore, and then you could follow up the hits. Using Biocore, we then confirmed that our fragments that came out of the biochemical assays actually came up as hits on Biocore as well. But the question is, where are these fragments binding, and how can we modify these fragments in a rational way in order to do proper drug discovery, or at least to move them from fragments to something that resembles leads or hits. So basically, I've just shown you here the profile of the binding affinity for particular hit, as well as K and ATP, and the fragment is shown here. The initial hit from the HTS is shown here at 18 micromolar with a KD of about 8 micromolar. So what did we do? So what we did was looking at what was out there. We didn't have any reliable crystal structures. So we started off using homology modeling, and this was a protocol that we came up with in order to try and answer some of the problems. So here in this protocol, it shows you that we'd come up with a homology model. We would use sitemap, which is a software that allows you to assess the druggability of a site. You'd use water maps to explore the solvent accessibility or polarity of the sites that we're going to target. And then you take our Biocore hits or our fragments, dock them using interaction fingerprints. And at the bottom here, I've got a list of publications which we have in the past used this particular methodology to try and accelerate discovery of hits. And so we're using this experience that we have in the past. We thought we'll be able to take the Biocore hits and propose some mutations within the active sites, or at least this would allow us to understand where our compounds were binding. In this way, we'll be able to quickly get to the where do we bind on the protein, and also hopefully give us an understanding of our binding modes for some of these fragments. So why did we choose to go for homology models? We chose to go for the homology model because the available crystal structure was poor in terms of its resolution, but however, it was also an apo structure. And in the apo structure, some of the active sites, especially the ATP active site, was occluded. So that was there was a loop that actually moves over the active site of the of ATP. So all the substrate binding sites were not usable in terms of the normal docking that you would do into a site. I'm just showing you here the problems that you had. So you had an occluded ATP site. You had a lot of missing and unresolved loops that were around the site itself, and some loop insertions which we didn't know how we could and where we could place these particular loops when you're actually doing some dockings. So how did we go around the problem? So we decided to do homology models based on the available analogs, but most of the available homologs were available from bacteria. So here we've got some PDB identifiers where we were able to get the templates for homology modeling. We did a bit of consensus homology model where we would graft the loops from the bacterial structures over the human structures and come up with a model that we thought would be a useful model for docking. We'll then dock all our fragments into the various sites that were available. So would we dock into the care site, or would we dock into the ATP site or other sites around the protein? So instead of just kind of doing random dockings, what we did was we used WaterMap to try and assess which one was the best site to go for, and sitemap as well. WaterMap is a way of kind of simulating the water molecules within the binding site. That way you'll be able to see whether this site is actually polar, very polar, or is available for being druggable. So basically, after doing this, we realized that not only did our fragments not dock into the other sites around the protein, especially the care site, but those other sites like the care site were extremely polar. And the only accessible site for fragments, and here are some of the fragments, the top hits, bound in our site or bound to the ATP site. While that was happening, we also tried to find out whether these fragments would actually be ATP competitive. Just because you can dock fragments into a site in a reasonable manner does not mean that you've actually found the site. So basically what we did was we did ATP binding competition assays on Biocore, where we would put ATP at the concentration, at the Km concentration, and then you'd bind the fragments. And barring none of our fragments, all our fragments lost activity in the presence of ATP. And within the organization, we also had the biochemical people working on trying to increase the concentration of ATP, of the fragment in the presence of ATP. And both the biochemical assay and Biocore assay confirmed that we have ATP competition. And so, looking at the ATP site itself and where our fragments were binding, we proposed some interesting mutations that you could use. So the idea was here we could use site-directed mutagenesis to trim away or to add or block the site with residues that would be interesting. So what we did here was, in this particular picture, I'm not allowed to show you the core fragment. We bound the fragment and realized that there was a potential pi-pi interaction between tyrosine 14 and most of our fragments, not all. We then trimmed that one to an alanine, and then we looked at all the other residues around the active site and trimmed those which we thought were interacting with our fragment to alanines. and ended up with some mutants that could be used to try and understand our binding mode. And here's a proposed binding mode on the side. So you've got the ribose pocket, where you've got the ribose pocket, you've got the ATP pocket, and the solvent accessible pocket. And these are some of the residues that were proposed by the docking to be directly interacting with our fragments. So using side-directed mutagenesis, we started probing the binding modes of our fragment. And in fact, after a little bit of protein engineering, and I sit next to somebody who's really keen at using mixed methodologies, we ended up generating mutants that were insensitive to our fragments. And I think I'm missing some of the graphics because they were in an animation here. But however, we also looked at other ATP sites of PAICS enzymes, in particular the chicken enzyme, which we knew was well expressed, and the E. coli enzyme, where we knew we had crystal structures. We then overlaid that onto our human enzyme and noticed that in the absence of tryptophan 14 and in the absence of leucine 33, we started losing activity in some of the species. So the chicken one, if we took YLPQ and we turned them to the chicken residues, we lost activity for our fragments. But when we grafted this back into the chicken, we started getting our activity back for our fragments. So here's some of the data. So in black is the human, in gray is the chicken. So basically, when we took a specific fragment, this unfortunately we've lost a graphic here, but fragment A, you would want to call it. That fragment A was at about 85 micromolar binding, activity for wild type. We took away tyrosine 14 and put it as an alanine. We dropped activity to about 570 micromolar, which was almost at the bottom of our assay. And then if we took Q and deleted it to an alanine, we noticed that we didn't have a loss of activity. But when we took the chicken in gray here and started grafting back these amino acids, we got a reasonable activity back for our fragments. And this was quite interesting, because as we started extending the fragments and the chemists started working on them, we noticed that as you went towards the tyrosine number 14 in the active site, as you grew your fragments in that direction, you actually started getting better affinity. But however, the picture where you trim this back to alanine, you lost all the activity. So this was giving us a feeling for where our fragments were binding and confirming some of the docking modes that we have. So I'm going to move on quickly here. So based on modeling and site-directed mutagenesis, we started looking at the emerging SAR and actually noticed that this particular binding mode was useful. And we presented that data to the chemists. And while that was happening, the chemists were working on the hits from the fragment screen. We also realized that some of the chemistries were difficult there, so we wanted to find alternative fragments. So here's where we went with this. So after doing all that mutagenesis study, we managed to show that our fragments bind to ATP. We managed to isolate which were the key residues for binding. We generated potential surrogate proteins for crystallography. So the chicken YLPQ protein may have been a better protein for crystallography, but so far that has not yielded results. And then we designed, based on these mutants that we came up, we also designed means and ways of trying to figure out whether we can get target engagement in a cellular system or not. So if you trim this back to alanines, you would be able to design a a construct that could be grafted into the cells of PAICS, which was functional but had lost all activity for had lost all binding for our fragments, and therefore our fragments wouldn't inhibit. And therefore you'll be able to see whether your fragments are actually driving phenotypes within a cell system or not. So after confirming where our fragments were binding, we then started looking at how to develop the fragments. So in terms of fragment development, what we did was we looked at all the dockings of our fragments, and here I'm giving you I've divided them into core 1, core 2, and core 3. And we looked at what were the best fragments in terms of their LE, which is a measure of lipophilicity activity. So the lower your LE, sorry, the higher your LE, the better. So we selected fragment 1 and fragment 2 based on the LE. Looking at fragment 1 and fragment 2, these were the binding modes that we came up with. And then we proposed that actually going towards water molecules that were generated by water simulation out here on the homology model would be useful rather than actually targeting the phosphate sites of the active site. So, however, chemists tried to do that, and we didn't get anywhere in terms of the fragments because of the intractability of these particular compounds. The chemistry was hard. We couldn't produce enough compounds to explore the SRR effectively. So one of the chemists came up to the modelers and said, look, what can we do in terms of, where else can we go with this? So what we did was we took all of Campbell, our MRC T library, and the PDB. We did a fingerprint similarity search of the fragments. We did a feature tree similarity search to find out whether there were scaffolds that we could hop onto. And we did crescent replacements where we wanted to look at the electrostatics around and shapes of the fragments rather than just looking at the topological similarity of the molecules. So using these methods, we then docked some of the results back into our fragments, and we realized that everything that we managed to get there was actually binding into the ATP site as well. But while we were doing that, we noticed that one of the Campbell fragments actually had a PDB structure, and this particular fragment had been discovered by another group who were working on ATP grasp enzymes. So we took a similar code to what was on that particular paper that had been discovered via the Campbell interface, and started grafting our ideas onto that particular core. And lo and behold, we managed to get from their fragment, which was actually completely inactive, to our fragment, which is designed with slightly different modifications to the core. And we ended up with things that were around about 150 micromolar for the human PAICS. I mean, you look at it and you go, oh, that's quite far. But this allowed the chemists to do a lot of chemistry around this particular moiety. And to cut the story short, within a measure of weeks to months, the chemists were able to, using the model that we had generated, propose some modifications to the theme, where if we put a halogen in this position here, we managed to get a boost coming from about 157 micromolar to about 10 micromolar. And that was a significant jump in terms of fragments. And however, exploring that solvent accessible pocket that we had characterized using water maps and simulation, we managed to get, just by modifying specific groups, we managed to get a jump from about 10 micromolar to about 100 nanomolar. And some of these compounds were then taken to the kinase profiling panel that we use to just check that if they're binding to an ATP site, will they also be hitting unrelated kinase activity? And these things were clean at a single point of 1 micromolar against about 84 kinases, human kinases. So we ended up with two alternative series, I suppose. The initial hit fragments and this follow-on fragment, which had been found via a literature. This particular fragment has now yielded compounds where we've further extended into the solvent accessible pocket. We've managed to get those compounds to come down from the 100 nanomolar to about 15 nanomolar. And these kind of activities were very encouraging. And this was done within, as soon as we got the right vectors, as soon as we understood where we were going in terms of where we could prod, it was almost impossible to get compounds that were that were not inactive. So my encouragement to those people who work with fragments and do not have structures is, try homology modeling. Try using the modeling in silico tools that you have at hand to at least suggest where you might be able to go with your fragments. So, I mean, I'm just going to summarize the results here. So we started off with fragments that were very difficult to synthesize, but these were around about 815 micromolar. Yeah, and we ended up with biaryl amide fragments that are related, which were around about 150 micromolar in activity. And this, using optimization, we're able to bring it down to about 15 nanomolar, to about 1500 nanomolar over a period of 2 to 3 months. And from then onwards, as soon as we could position our fragments and we were happy with our models, we were able to drive that activity to single-digit nanomolar within months. And I also encourage you to go and participate, or if you're going to this particular conference in Vienna, these fragments will at least most of the chemistry and the full chemistry story will be given at that talk. And thank you very much for listening. I hope that the interference has not put a lot of people off, and that if you've got any questions, please do come back to me. And I encourage you to write me an email, and if you've got any further questions outside of the particular talk, to come out and we can discuss how we managed to turn this particular challenging project around. Thank you.
Thank you very much, Chido. It was, I think, good that we converted to PDF in the end, so then it went on very smoothly. Thanks very much. Dear audience, in case you have any questions that pop up to your mind, also just raise your hand in the GoToMeeting control so you can speak out actually, or just type into the chat box so that we can record your questions or consolidate and then ask Chido directly here. While this is happening, perhaps I just kick off with an old question that I have. Chido, you mentioned LE as ligand efficiency. Here and there again, of course, the UK has a big tradition in using these indices. Looking back in this project, would you esteem this as an additional worthwhile guide, or is it considered as a filter for you, or how do you actually take this along or not? Could you comment on that?
I think in this particular project, it was a way of, because we had, in the beginning, we had something like 6 or 7 different cores in terms of the fragments that we had.
Excuse me, Chido. Excuse me to interrupt you, because one person is just jumping in. What does LE stand for? If you could just do a little loop on this, so that people gather.
So the LE is it's a calculation to give you a feeling for. Let's say it captures your affinity relative to the lipophilicity of your compound. So you want to be able to say, if a compound is very active, the activity is not being driven by just simply that particular compound being lipophilic, being very lipophilic. Yeah. So you kind of we use C log P in order to measure our kind of because at this point we didn't have we used calculated properties in the beginning, but as we went along, we managed to measure the log P and the C log P of the fragment. So you could measure how much activity do I get from grease, as it were. Yeah.
Right. Okay. So back to that question. Thanks for that.
So the question you were asking is why would we have used LE, and did it make any difference?
Yeah, was it a guideline? Was it a filter or something like this along those lines?
I think in a sense it was more of a filter because we had enough fragments to work on, and we did not have enough hands to work on those fragments. We really wanted a fragment that we thought had better properties. And this was one way of just measuring. Are we getting affinity because of just grease, or are we getting affinity because because of some reasonable polar interactions?
Mm-hmm. I see. Yeah. Thanks very much. Looking with half an eye onto the list of further questions. So don't be shy, guys. In case there is more that you'd like to know, just feel free to type in or raise your hand. There is, I think, one question from Bertrand Leblanc. I'm just trying to demute him in case that's a question, but he also commented on the ligand efficiency point himself. Bertrand, if you want to speak up, feel free to. You're on.
No, just to say that ligand efficiency is LE.
That's right. Yeah, it's the L is ligand and E is efficiency. Okay, cool. Thank you. Thank you very much, Bertrand. Okay, so appraisal here and there. Andrea Tagliani, who typed in, nice work, Chido, thanks. Turning point. Well, the story was, in my opinion, the ATP antagonism. Never thought of an allosteric sort of ATP antagonism. Do you have any proof now to exclude allosteric mechanisms? That's a good question from Andrea. Joe Andrea from this side. Yeah. Did you comment?
That's a good question. I mean, from the fragment hits that we got, it was quite clear when you look at the actual structures themselves that you could suspect that there were ATP competitors. Looking at the dockings as well as the fragment matter that we got, I think it was clear to us that we are actually competing with ATP. When we then did the ATP competition, which was kind of flowing ATP at a steady concentration on the biocore, so that when you did introduce your compound, you lost all the binding from the particular compound, so you lost all binding. When I showed a slide where you had ATP itself bound, and we thought, and actually yes, in the beginning we didn't know whether this was being driven by an allosteric or it was actually ATP competition, but there is no doubt now that these things are ATP competitors. I don't know if I've understood the question and I've answered it properly.
Andrea, I just unmuted you, so in case you would like to comment, feel free to speak out. Okay. And Andrea does not want to speak out loud, but perhaps he wants to type in. There is fine to me that he writes. Okay, cool. Is it general or was that specific for this particular example or ligand? Mohamed Ayaz Anwar asks, but actually now this question is a little bit hard to put into context. So I will also unmute Mohamed. So in case you would like to speak out, Muhammad, and make this question a little clearer, then feel free. You are unmuted now. Muhammad?
Hi, this is Muhammad. Can you hear me?
Yes, we can hear you loud and clearly.
Okay, thank you. Dr. Chido said that higher L is better. I'm just want to ask that whether higher L is better for this particular case or it is general.
Well, I, because of what Amy is trying to tell you, we think that what you're trying to do.
Audio is bad.
Can you,
did you change anything? Could you repeat and perhaps check that the microphone is sitting okay? No, we don't hear you.
I'm just check.
Yeah, now you're good.
Am I okay now?
Yes, absolutely.
Okay, so I was just responding to this. I mean, it's not an absolute. There's no absolute value that you would say at this point, this is the cutoff for a good LE or a bad LE. It's more a way of ranking, so that when you're doing your ranking, you're ranking it in such a way that that the ones that have got a higher LE go to the top. Yeah. So it's just a way of flagging out compounds that are gaining a lot of affinity or a lot of binding affinity because of grease. You don't want those compounds. You want compounds that are forming, especially when you're looking at fragments. You don't want just a blob of grease. You want something that actually is binding. But this was, there's no absolute value that you can derive from LE.
Okay, so for the time being, it doesn't seem that there are any further questions popping up, and that's pretty perfect timing, actually. So 1 minute to go. With this, I'd like to thank you, Chido, one more time, and I will take back moderatorship. So I would like to make you save the next date for the next webinar, which will be in the new year, end of January 25th, 2007. At the same time, 3 p.m. Berlin time, that means 2 p.m. London time, 3 p.m. minus 6 hours is Boston, New York time, minus 9 hours Los Angeles, plus 3 or 4, I think, depending on where you are, India, and so on and so forth. And we have one of the very well-known guys around who knows all about LE and lipophilic LE, etc. Marcel Verdonk from Aztecs. Actually, we're still pondering with him about the title, but I've seen him many times, and I can promise it will be an exciting webinar. So watch this space.